Hello everyone, this is Dr. Tarun Kumar Patel, Assistant Professor Biotechnology from Sant Guru Ghasidas Government PG College, Kurud, District Dhamtari, Chhattisgarh. In today's lecture, we will discuss about publicly available tools. This topic is useful for MSc Biotechnology second semester, paper 5th, Biostatistics, Bioinformatics and Computers in Biotechnology, Unit 4. Let's see. First, the introduction. Extracting any meaningful information from the vast amount of available data requires some specific tools. Here we are talking about the biological data, which might be related with the proteins, nucleic acid, or other molecules, or they might be the data related to some molecular structures. This data would be further used for extracting some other information like the structure of any compound, like their interaction, like their three-dimensional configuration. All these thing can be performed using some specific tools, or we can say softwares. These tools or softwares can be categorized in two different groups. First, the publicly available tools, which are lawfully made available to the general public, whereas some tools are not readily available for the general public, but they have some restriction. They might have some um, licensing fee or any other restrictions. So here we will discuss about publicly available tools which are freely available for everyone to be utilized for processing the biological data. Basically, there are a number of tools available which are publicly available for the database analysis. But here our focus is the tools which are basically available at NCBI. However, there are some tools we will discuss about the tools which are not available in NCBI, but they are mostly very much useful tools. So let's see these tools one by one. Thousand Genome Browser. The Thousand Genome Browser is an interactive graphical viewer that allows user to explore variant calls, genotype calls, and supporting evidences such as aligned sequence reads that have been produced by the Thousand Genome Project. Amino Acid Explorer. This tool allows user to explore the characteristics of amino acids by comparing their structural and chemical properties, predicting protein sequence changes caused by mutations, viewing common substitutions, and browsing the functions of given residues in conserved domains. Basic local alignment says tool or BLAST. This is one of the highly used tool available at NCBI. The BLAST finds regions of local similarity between biological sequences. The program compares nucleotide or the protein sequence to sequence databases and calculate the statistical significance of matches. BLAST can be used to infer functional and evolutionary relationship between sequences as well as to help identify members of gene family. CD-Tree, a standalone application for classifying protein sequences and investigating their evolutionary relationships. CD-Tree can import, analyze, and update existing conserved domains or CDDs, records, and hierarchies, and also allows user to create their own. CD-Tree is tightly integrated with Intras, CDD, and CN3D, and it allows user to create and update protein domain alignment. Cobalt. Cobalt is a protein multiple sequence alignment tool that finds a collection of pairwise constraints derived from conserved domain database, protein motif database, and sequence similarity using RPS BLAST, BLAST-P, and PHI BLAST. CN3D. CN3D is a standalone application for viewing three-dimensional structures from NCBI's Interest Retrieval Service. CN3D runs on Windows, Macintosh, and Unix and can be configured to receive data from most popular web browsers. CN3D simultaneously displays structure, sequence, and alignment, and has powerful annotation and alignment editing feature. Conserved Domain Architecture Retrieval Tool, or CDART. It displays the functional domain that make up a given protein sequence. It lists proteins with similar domain architectures and can retrieve proteins that contain particular combinations of domains. Conserved Domain Search Service, or CDSearch. It identifies the conserved domains present in a protein sequence. CDSearch uses RPS BLAST, reverse position specific BLAST, to compare a query sequence against position specific score matrices that have been prepared from conserved domain alignment present in the conserved domain database, or CDD. Genome Data Viewer, or GDV. A genome browser. It is basically a genome browser for interactive navigation of eukaryotic reference sequence genome assemblies with comprehensive inspection of gene, expression, variation, and other annotations. GDV offers easy to load analytical track pre-configuration, a menu of data tracks for easy display and customization, and supports upload and analysis of user data. This browser also enables the production of displays for publishing. Genome Remapping Service. NCBI's Remap tool allows user to project annotation data and convert locations of features from one genome assembly to another or to a reference sequence gene sequences through a base by base analysis. Options are provided to adjust the stringency of remapping, and summary results are displayed on the web page. Genome Workbench, an integrated application for viewing and analyzing sequence data with Genome Workbench. Anyone can view data in publicly available sequence databases at NCBI and mix this data with someone's own data. Multiple Sequence Alignment Viewer. It is an integrative web application that enables users to visualize multiple alignments created by database search results or other software applications. The multiple sequence alignment viewer allows users to upload an alignment and set a master sequence, and to explore the data using features such as zooming and changing of coloration. OSIRIS, a public domain quality assurance software package that facilitates the assessment of multiplex short tandem repeats DNA profiles based on laboratory-specific protocol. OSIRIS evaluates the raw electrophoresis data using an independently derived mathematically based sizing algorithm. It offers two new peak quality measures: fit level and sizing residual. It can be customized to accommodate laboratory-specific signatures such as background noise setting, customized naming conventions, and additional internal laboratory controls. Open Reading Frame Finder or ORF Finder. It is a graphical analysis tool that finds all open reading frames in a user's sequence or in a sequence already in the database. 16 different genetic code can be used, and the deduced amino acid sequence can be saved in various formats and searched against protein database using BLAST. Phenotype Genotype Integrator. It supports finding human phenotype or genotype relationships with queries by phenotype, chromosome location, gene, and SNP identifiers. Currently includes information from DataBackGap, the NHGRI GWAS catalog, and GTAX displays results on the genome on sequence or in tables for download. Primer Blast. The Primer Blast tool uses Primer 3 to design PCR primers to a sequence template. The potential products are then automatically analyzed with a BLAST search against user-specified databases to check the specificity to the target intended. ProSplegion. ProSplegion is a utility for computing alignment of proteins to genomic nucleotide sequences. It is based on a variation of the Needleman-Wunsch global alignment algorithm and specifically account for introns and splice signals. Due to this algorithm, ProSplegion is accurate in determining splice sites and tolerant to sequencing errors. PubChem Power User Gateway or PUG, PUG. PUG provides access to PubChem services via a programmatic interface. PUG allows user to download data, initiate chemical structure searches, standardize chemical structures, and interact with the e-utilities. PUG can be accessed using either the standard URLs or via SOAP. PubChem Structure Search. PubChem Structure Search allows the PubChem compound database to be queried by chemical structure or chemical structure pattern. The PubChem catcher allows a query to be drawn manually. Users may also specify the structural query inputs by PubChem compound identifier or CID information. Smalls, smarts, in shell molecular formulas, or by upload of a supported structure file format. Related structures. The related structures tool allows user to find 3D structure from the molecular modeling database that are similar in sequence to a query protein. Although the query proteins may not yet have a resolved structure, the 3D shape of the similar protein sequence can shed light on the putative shape and biological function of the query protein. Splegion, a utility for computing cDNA to genomic sequence alignment. It is based on a variation of the Needleman-Wunsch global alignment algorithm and specifically accounts for introns and splice signal. Due to this algorithm, Splegion is accurate in determining splice site and tolerant to sequencing errors. TreeViewer. TreeViewer is a tool for creating and displaying phylogenetic tree data. TreeViewer enables analysis of your own sequence data and produces printable vector images as PDFs and can be embedded in a web page. Variation Viewer. Variation Viewer is a genomic browser to search and view genomic variation listed in SNP's database, variation database, and clinical variation databases. Searches can be performed using chromosomal location, gene symbol, phenotype, or variation IDs from SNP databases or variation databases. The browser enables exploration of results in a dynamic graphical sequence viewer with annotated tables of variations. VecScreen. VecScreen is a system for quickly identifying segments of a nucleic acid sequence that may be of vector origin. VecScreen searches a query sequence for segments that match any sequence in a specialized non-redundant vector database. Vector Alignment Search Tool or VAST. It is a computer algorithm that identifies similar protein three-dimensional structures. Structure neighbors for every structure in MMDB are pre-computed and accessible via links on the MMDB structure summary pages. These neighbors can be used to identify distant homologies that cannot be recognized by sequence comparison alone. And the last one is viral genotyping tool. This tool helps to identify the genotypes of a viral sequence. A window is slid along the query sequence and each window is compared by BLAST to each of the reference sequence for a particular virus. Then there are some tools from the other sources other than NCBI. The first one is GROMACS. GROMACS, it is a very versatile package to perform molecular dynamics. It means it simulates the Newtonian equations of the motion for systems with hundreds to millions of particles. It is primarily designed for biochemical molecules like protein, lipids, and nucleic acid that have a lot of complicated bonded interaction. But since GROMACS is extremely fast at calculating the non-bonded interaction that usually dominate simulations, many groups are also using it for research on non-biological systems like in polymers. GROMACS supports all the usual algorithm you expect from a modern molecular dynamic implementation. Then RASMOL. RASMOL is a computer program written for molecular graphics visualization intended and used mainly to depict and explore biological macromolecules structure, such as those found in the protein data bank. It was originally developed by Roger Saile in the early 1990s. The next one is Modeler. Modeler is used for homology or comparative modeling of protein three-dimensional structures. The user provides an alignment of a sequence to be modeled with known related structure, and a modeler automatically calculates a model containing all non-hydrogen atoms. Modeler implements comparative protein structure modeling by satisfaction of spatial restraints and can perform many additional tasks, including de novo modeling of loops in protein structures, optimization of various models of protein structure with respect to a flexibly defined objective function, multiple alignment of protein sequences and or their structures, clustering, searching of sequence databases, comparison of protein structure, etc. Modeler is available for download for most Unix, Linux system, Windows, and Macintosh. Was originally written and is currently maintained by Andres Sally at the University of California, San Francisco. It runs on operating system Unix, Linux, Mac operating system, and Windows. It is a freeware for academic use. Ensembl. Ensembl is a genome browser for vertebrate genomes that supports research in comparative genomics, evolution, sequence variation, and transcriptional regulation. Ensembl annotate genes, compute multiple alignment, predict regulatory function, and collect disease data. Ensembl tools include BLAST, BLAT, BioMart, and the Variant Effect Predictor or VEP for all supported species. Clustal Omega is a new multiple sequence alignment program that uses seeded guide trees and HMM profile techniques to generate alignments between three or more sequences. And the last one here is FASTA. FASTA is another commonly used sequence similarity search tool which uses heuristics for fast local alignment searching. So these are the tools which are publicly available. Let's see the conclusion. So we have already seen a variety of publicly available bioinformatic tools which are available and which are useful to carry out sequence or structural analysis, study of molecular dynamics, or extracting or representation of data in more meaningful form. Thank you.